Okay. All right, welcome back, everybody. We are starting the second short talk session for today. A reminder for those of you online, if you have any questions, please put them in the chat or the Q&A. We check both places. We have 8 minutes talks. I will set a timer and I will wave to you 1 minute when it's done. So please keep an eye on me. I'll try to sit over there so you can see, and then you will get the if you hit 8 minutes. We're trying to keep on time. Also, it's fun. Okay, so our first speaker up is Mengbo Li, and she'll be talking about cell type specific microenvironment analysis on spatial transcriptomics data.
Hello, everyone. My name is Mengbo, and I'm from WeHi in Melbourne. Today, I'd like to give a brief. Am I time up? Today, I'd like to give a very brief overview of CIDR, which is an R package we've recently developed, which performs cell type specific microenvironment analysis on spatial transcriptomics data. So CIDR stands for Spatial Cell Type Intercorrelation by Density in R, and our aim is to perform cell type specific microenvironment analysis on spatial transcriptomics data. But then one of the most important question is that how do we even define the microenvironment using the spatial information we have available? So here, let's have a look at a real data set. It's a public MERSCOPE data from human breast cancer tissue. So here we have already performed the cell type annotation using single R. And so to start with, we can ask ourselves the very simple question of what do we observe from this annotated data set, from this spatial map of this. So here, each point is a cell. So what do we observe from this slide? So when we can see that cancer cells, which is in light green, is continuously spreading over the entire slide. And macrophage cells, which is in the dark blue, is they are forming small clusters embedded in the cancer tissue. And so on. There are many other observations. And we can then translate these empirical observations into the mathematical terms, which means that what we see means different cell types have different spatial density patterns. And really, the intuition behind CIDR is that we can then define a cellular microenvironment using this spatial density patterns, which is a very simple but completely normal approach. So, for example, here I have calculated the spatial density patterns, the spatial density function for cancer cells. And here on the left-hand side is the contour plots, where the red correspond to the higher density levels of cancer cell, and the blue corresponds to the lower density levels. So we can, CIDR then automatically detects region of interest using network analysis approaches and through this find ROI function. So here in this example, we have identified 11 different regions of interest. Each corresponds to these regions that are dense in cancer cells. In the meantime, CIDR is also the ROI detection is also highly easily tunable by the users. So say, for example, after talking to our favorite histologists, we believe that this slide is actually coming from a very homogeneous cancer tissue. So we can then probably relax the filtering on the density level a little bit, and that would give us this one big ROI, which we'll base our further analysis on. So we can then overlay the region of interest with the contour plot we have just estimated. And then this would allow us to calculate the cell proportions at each level of cancer density. So this would give us the cell type composition plot on the right-hand side, where, for example, we can see that fibroblasts in pink is decreasing in proportion, and also epithelial cells, which is in orange, is increasing in proportion as the cancer density increases. And next, we can also calculate the spatial density of other cell types in this same region of interest, and and calculate the correlation between the spatial densities of different cell types. And that would actually be the cell type co-localization analysis, or sometimes also called the neighborhood analysis. So here, for example, we see that epithelial cells and the cancer cells, they show a positive correlation in their spatial density patterns, which means that epithelial cells and cancer cells tend to co-localize in this region of interest. And as another example, fibroblasts, there is a negative correlation between fibroblast and cancer cell densities, which means that these two cell types tend to not co-localize. And following that, we can also perform D analysis using pseudobulking. So to construct the pseudobulk samples, we can, cells are grouped by the cell types at each cancer cell density level. And here I have constructed the pseudo samples for fibroblasts. And here in the MDS plot, each point is a pseudo sample, and they're labeled by the cancer density level where the fibroblast cells are from. So we can already see a very clear trajectory in this MDS plot among the pseudo samples according to the cancer density levels. And then to perform the actual D analysis, we can use HR pipelines. So for example, so here, let's say we're interested in answering the question of how does gene expression change in fibroblasts as cancer cell density increases. So we can answer that by constructing a design matrix using the natural cubic spline transformation, which models the relationship between gene expression and the cancer density level. And then to perform the DE tests, we can use the quasi-likelihood F-test pipeline to test if this trend or this relationship between gene expression and the cancer density level is significant. So for this example, we have identified 284 genes that are significantly differentially expressed in fibroblasts as the cancer cell density level increases. And in particular, I want to point out that the first column, the first coefficient, represents the linear relationship between gene expression and the cancer's density level. And here we can also visualize the trend, this natural cubic spline trend. Say, for example, on the right-hand side, I have for the top one DE gene, we can see that CXL12 expression decreases in the fibroblasts that are located in the cancer-dense regions. So in summary, we introduced CIDR, which performs cell type-specific microenvironment analysis for spatial transcriptomics data. So one of the key features is that CIDR defines regions of interest based on the spatial density of each cell type. So today, I'm only giving a very high-level overview of the methods, but happy to chat further if there are any questions. So our package is already available on Bioconductor. So it takes the spatial experiment objects as input, and we've made it compatible with all current HR and lemma pipelines for downstream analysis, such as differential expression and pathway analysis. And with that, I'd like to thank my supervisor, Andy, and also our collaborator, Ning from Sigency. Thank you.
Yeah, that's super cool. Great talk.
Thank you.
Yeah, just one question when it comes to cell density. So different cell types can have very different sizes, and apparently here, I think you're using the centroids to compute cell density.
That's correct.
Would you consider correcting cell density, adjusting cell density based on cell size? Extreme example, adipocytes are very large compared to leukocytes.
Yes, so that is a great question. So I think so we have definitely looked into that, but then so one of the backbone of our method is the spatial density function estimation. And that typically involves some smoothing, like kernel density smoothing. And it actually, so something that I didn't touch today is that the grid size we do on the smoothing actually has some impact on the results. But then after some experimenting, we've decided that intermediate to larger smoothing actually would improve the analysis. So the cells, so it's like, so if you go into that high resolution, we would enter subcell analysis, and that would actually make your spatial density function not easy to work with and actually quite challenging.
This is really fantastic work. My question is, how translatable are these densities between samples? And then at those same densities, how much do the cells within those densities agree across samples in terms of their expression?
Yeah, that depends, excuse me. So that depends on whether you have replicates.
Yeah.
Right. So I think what we believe that, well, at least I believe that each slide, no matter how close they were sectioned from, they are different samples. And if like the spatial density function is just like an estimating the patterns based on the slide you have and based up and from and also after cell type annotation. So really it, like the estimation itself doesn't have a big impact. It's mostly like even you can like investigate very like visually are these two slides similar? If they are similar, then the estimation would be similar.
I mean in the context of separate tumors, entirely separate, not even different slides from the same tumor, but if you look at separate tumors, do you see agreement in terms of actually the cellular density tells you something biologically about what's happening?
Yes.
Okay.
And this is also another key feature that I didn't talk about is like our current framework with ROI and stuff and ROI and contour levels, it's allowing us actually to include bioreplicates into your DE analysis procedure.
That's really awesome. Cool. Thanks.
Thank you.
All right. So our next speaker is Lukas Weber, who will be talking about identification of spatial domains by smoothing for compositional analysis in spatial transcriptomics data.
Thank you. So I'm Lukas Weber. I'm now at Boston University. I'm talking about a method called smoothclust for identifying spatial domains in spatial transcriptomics data. So spatial transcriptomics again. So we're measuring transcriptome scale gene expression. And in this example on the right, in this work, we're mainly focusing on Visium data right now. So that's a spot-based technology where we're measuring at a grid of spatial locations shown there. And our data looks like this. So we have these gene by cell and cell or spatial location gene expression count tables. And in the case of spatial transcriptomics, we also have the spatial coordinates to work with and possibly image features depending on what we've captured. And in a typical analysis workflow for this type of data, we have several different steps. So starting with pre-processing and then further downstream analyses, feature selection, dimensionality reduction, clustering, and so on. And here we're focusing on clustering to identify spatial domains or spatially distributed cell populations. And some examples there on the right from different data sets. And specifically in unsupervised clustering, we want to classify measurements, cells or spots into groups without relying on any external labels, so using an unsupervised algorithm. And we want to distinguish here also between cell type clusters and spatial domains. So if we're measuring cell type clusters, then we're just using the gene expression measurements, whereas with spatial domains, we're also going to the spatial coordinates and further analysis. And we can also distinguish our algorithms as spatially aware or non-spatial clustering algorithms. Now in smoothclust, we want to segment regions of tissue areas into spatial domains with smooth spatial boundaries using the gene expression measurements and spatial coordinates as the inputs. And we want to use this as a pre-processing step for further downstream analyses at the spatial domain level, such as cell time compositional analyses or pseudobulk differential expression. And we're not necessarily assuming that cell types are the same as spatial domains here. And so we really want these smooth spatial boundaries we can see in this example here. And the algorithm is quite simple. So at each spatial location, we're calculating average gene expression at each spatial location across neighboring spots using some definition of neighbors. And we have some design choices here, such as the method to select the neighbors, bandwidth parameters or smoothing parameters, and optionally weighting scheme for averaging. And in the default implementation, we're simply using a circular window with an adjustable bandwidth parameter, by default 5% of the width or height of the tissue area, and no distance weights for averaging. And that gives us then, as an output, spatially smooth gene expression values, which smooths out both sampling variability and sparsity at the at the gene expression count level, and also biological variability in cell type mixtures per spot, which we have especially in the Visium data. And these smooth values are then uniform within regions consisting of consistent cell type mixtures. And we also have several alternative implementations, including a kernel method, where we're calculating a distance-weighted average within a circular sliding window with a truncated exponential kernel, but there's a computational trade-off there, where the uniform method works much faster. And now we've run this on several different data sets. So here, this is the human breast cancer data set with Visium from 10x Genomics. And we see here, so if we do standard clustering using just the gene expression measurements, then we have these sort of mixed regions with different cell type clusters, but with mixed cells per spot. So if we want these smooth regions, this is what we get with smooth clusters as an example in this data set, using the uniform method, bandwidth 5% of the width or height of the tissue area. And so we get these really nice smooth areas and can use these as pre-processing steps for further downstream analyses at that spatial domain level. And then to quantify this level of smoothness, we have this metric we've developed. So the number of neighbors per spot from a different cluster ID. So this can be used to quantify how smooth these boundaries really are. So here on the left, comparing the non-spatial clustering and the spatial clustering on the right, where it's much smoother. And then we can use this as the input for, as an example here, compositional analyses of cell type clusters per spatial domain, and then also pseudobulk differential expression testing at that spatial domain level. Different data set here. This is in Xenium, so Xenium mouse brain, so single cell spatial resolution now, 248 genes in this data set. And if we're applying that, then again, we get these nice smooth regions, this example here on the right, with 10 clusters in this case. And the package is now in Bioconductor, smooth cluster with a tutorial as well, with some examples. And we're working on a preprint and additional data sets. And with that, I'd like to thank my current and former collaborators who've been working on this, and in time for any questions.
This is a really nice way to be able to kind of look at your visual data in the spatial context and determine biological differences, but are you able to control for global differences across biological replicates?
So that's the next step. So right now we're working on new data sets with multiple samples, and that's challenging, but we're working on that.
Gotcha. Thanks.
Can you say a little bit about why the region should be smooth? Or is there a tuning problem where you might have, pods reaching out that shouldn't be smooth?
So it depends on the biological context, yes. So if it's a context where we want smooth regions, then yes. So if we're making that assumption that we want that for purposes of interpretability. But in some contexts, if that's not appropriate, then that wouldn't work.
Okay, thanks. Joining. And okay, I will just say, Mengbo, you had a couple of questions online that we didn't have time for, but we'll see if we can get you to answer them at some point. All right. Okay, our next speaker is Scalable Count-Based Models for Unsupervised Detection of Spatially Variable Genes by Boyi Guo. Did I say that okay?
Hi everyone, my name is Boyi. I'm really excited to be here to share some a work in progress. So I think previous speakers did a introduction for spatial transcriptomics, so I'm not gonna do that. Instead of doing that, I'm gonna invite you to a time travel. So please bear with me. And let's go back to 2013, where everybody are excited about this novel data type called single cell RNA-seq. And then how we're going to model it, I think that is the question we asked ourselves. And then I guess the first thing we want to try is that let's work out something, or let's follow the same thing we did for the box single cell data analysis, which is including normalization or log transformation of normalized data. And then a couple years passed on, and then there's more people realize that log transformation may not be a good idea. And then by one of the paper I show here is that you can see when we do the log transformation on a normalized measurement, then there's access zero inflation are introduced to the data set. And then more importantly, if you use the same log transform data to do a PCA on it, then you can see this artificial clusters in a data set that doesn't contain any biological signal in it. However, if you model the data without the log transformation based on more generalized frameworks, such as generalized linear model with the PCA, then those artificial signals are gone. Now, let's travel back to current dates. Now we have the spatial resolved transcriptomics. We're hyped about it. And then we still ask the same question, how do we model it, right? So then there's, so this is coming back to the spatial transcriptomics, where we can actually map the gene expression in a more spatial context, and then using it as a way to quantify or analyze or understanding the molecular signature of functional regions or anatomical regions. And also, as Lukas mentioned, it also serves as a step of feature selection. But mathematically speaking, is that now you have a count matrix, and then you also have additional spatial coordinates. And the principle is you want to find the association between these two factors. And then this is actually a well-studied, like if we're only just looking at this data format, it's actually well-studied in statistics history, which one of the easy way to do it is using Gaussian process, right? So you assume that the data collected within a quite close distance are correlated together. So you essentially use a covariance matrix to capture the spatial expression where using some different kernels. And then here's a visualization how you can use different kernels or different specification to capture that spatial variation. However, the challenge here is that we actually have const data. And then do people want to fit a const data to a normal distribution? Probably not, right? Most people probably not going to try that. Now we just like try to normalize it or using this log transformation. And then wait a moment, we just say that log transformation is not that good, right? Mind-blowing. So in previous, last year, there's a graduate student, Kinry Shah, in our group actually working on trying to see what is actually downstream impact. And then in her talk, which is last year in Bioconductor, we saw there's some artificial relationship between the marginal mean of each gene and also the rank based on some Gaussian process model using the log transformation data as input. And then there's a way to address it, which is very inspired by the classical model WOMB, which trying to use the mean-variance relationship to calibrate the process. However, we also, like, there's some other challenge in this process. And then we ask ourselves, can we actually model the count data as is? And there's some work of it, which is brilliant. However, there's a challenge, practicality or scalability. This nice computation framework use 10 seconds per gene. And then at the time, the data sets only have 250 observations. So it takes forever. It's basically like if you start running the program before Bioconductor started, and then at the end of Bioconductor, you can check your results. So scalability is a threat in the data analysis as the data getting larger and larger. So here I'm actually proposing to repurpose a really fantastic framework from generalized additive model, specifically this book or a package from Simon Wood. And then I'm using the simplate spline to model the gene expression in the space. And then for people who don't know spline, the basic idea is you connect dots together and then form a smooth version of it. And then the nice thing is that you can formulate in a more linear combinations and then feeding it back into the generalized linear model framework, which we know is very scalable. So the basic idea is we start to use spatial coordinates back into this spline format, and then using that to model the counts or normalized counts. So the nice thing is we can leverage this framework and then model the counts data as Poisson, adjusting for some scaling factors, regardless what you deem appropriate. And also in this case, you can actually formulate the spline or the spatial coordinate or the spatial surface as a fixed effect or a linear combination, and then just leverage generalized linear model framework. And then you can also open up to other distribution assumption on the data generation. So if you like connected binomial, you can do that. There's also exponential family, and then back into, this is part that I haven't extended, but you can also leverage the MGCV package where they also consider zero inflation if you really believe in that. And also this is a quick result showing that if we try to use the Poisson distribution, and then which is shown in the grain, then you don't see this artificial mean rank relationship there. And then we also desimulation trying to see the rank estimation under different data generating mechanisms. And we see using the thin plate spline with Poisson, it actually improves the rank estimation. And also it's scalable. So I think the thing that I'm actually interested in is leveraging the framework, the generalizability model, to understanding why, how log transformation is changing or ultra processing the data. And then this could be, you can see if we fit the same data, same model with different data as input, you can have different surface, and then that will have downstream impact on your spatial domain estimations. And then I just want to end up my talk with this metaphorism. In my mind, I think cons data is just grain. It's kind of like too raw to process. And then the normalized data is kind of like you make it a flower, then you can just do things as your own. And then the log transformation, sometimes it's just take the flower and make a cookie. Me personally, I don't hate cookie. I enjoy cookie from time to time. But the question I want to ask you is, do you want to know how the cookie being made? And then do you want to consume the cookie every single day? Thank you. Yep. And also, I never time travel, so I don't know what background music I should put on there. So if anybody know how to time travel, tell me how that feels, what sound it is. Next time, I'll incorporate that to my talk. Yeah, I'll think about that. Thank you, everyone. Any questions?
We're missing online questions. Okay.
Sorry. So is there a particular reason why you chose generalized thin plate spline? Is there sensitivity analysis regarding this?
This is a great question. I think thin plate spline, there's unique property. I think it's called isotropic, meaning that it scales well on the two dimensions. It just match the physical representation of data we collected. That's one of the reason. And also, I think there's more complicated two-dimensional splines frameworks, such like soap film, that address more complicated questions, but the scalability there is not as good as thin plate. And then the thin plate is Simon would have a really brilliant way to estimate the thin plate to a lower dimension, lower rank representation. So this is why the framework is actually scalable. If you just try the original really OG thin plate, it doesn't scale that well actually. So this is my reason why I prefer to do this.
Thank you.
Yep, thank you.
Thank Boyi again.
Thank you so much.
You got an amazing talk. Note online. Okay. All right. Our next speaker is Michael Totty, who'll be talking about SpotSweeper, spatially aware quality control for spatial transcriptomics.
All right, hi everyone. My name is Michael. I'm a postdoc fellow at Johns Hopkins. Yeah, today I'm going to be presenting my first BioC package focused on QC for SRT technologies. So I won't go into too much detail about the background. I know we've already heard a good amount about it, but most of the data I'm going to show today, and this package right now is focused on spot-based or sequence-based methods. However, regardless of this, what the package is currently able to do, regardless of what technology you want to use for SRT, there's this common truth that people are starting to realize is that a lot of the QC features, including the library size, is biased by the spatial biology in these slices. So these are some images from a recent paper showing the library size for 5 different technologies, including spot-based as well as imaging-based technologies. You can see the clear spatial patterns in the library size. And so this paper is really focused on kind of what Boyi was just talking about when you go to normalize the data, which is of course a very important problem. But however, the library size is also very important for QC. So currently the QC methods for SRT technologies are really just adopted from the single cell methods. And this is a really simple idea where you treat every single spot as if it's a cell, and then you remove the spots with a low number of total UMI counts compared to all other spots in that sample. And so it would look something like this. We have a violin plot that shows every single sample, and then essentially the outliers on the lower end, you would just pull those out as if they are bad or low quality spots, right? However, the issue here is that unlike single cell, where you are sampling a pretty uniform part of tissue for every single observation, so either a nucleus or a cell in every single observation. In SRT, depending on what is underlying, in this case, the spot, it's going to be very different. It could be a spot of tissue that has 0 cell bodies or 0 nuclei, or it could have 4 or 5 cells. And so the library size is going to be different because of the biology that underlies this. And so if you just globally go in and cut out the spots that have lower library size, you can see where you have biases. You're actually going to be removing what is true biological signal from your data. And so sure enough, if we do this, we can see here, this is a piece of, this is a Visium data set from the cortex in humans showing these 6 different layers here, as well as white matter and black. And so within these layers, there are different densities of cells. And so layer 1 and white matter in particular have very low or even no cell bodies at all. So you would expect the library size to be much smaller in these layers. And so if you just globally exclude cells based on the library size, you would expect to exclude a lot of these spots. And sure enough, you can see that there's obvious bias here in the spots that are deemed low quality and removed. You're going to remove a lot of your white matter as well as your layer 1 in these samples. And so that's why we wanted to develop a method that can do QC a bit better, help you retain your data, but also still identify the true low quality spots. So this is the rough outline for SpotSweeper. It's a very simple method using the standard QC metrics that everyone already uses, which is library size, unique genes, and the mitotic percent. We, instead of comparing every spot to the global sample, we compare it to its local neighborhood instead. So we use KNN, we find the neighborhood for every spot. We then normalize it based on a z-score, and then you can do your outlier detection based on that transformed z-score data, essentially. There's also another module where we can detect larger artifacts, so I'll talk about that in the second half of the talk. So going back to this example sample here, again, we can see that using the original methods for single cell, we're excluding a lot of biases based on layer, but using SpotSweeper and the local outlier detection, we can see that now we remove a lot of that bias. So importantly, there's 2 things here. We are no longer removing these high quality spots that were biased based on just the lower library size. We are still removing a lot of the same spots that were detected, and these are true outliers. We can scale this up and look at it also across a lot of samples. So this is showing the distribution across 12 different samples here for each of the layers here. And you can see that there's quite a lot of bias within these layers. So if you were to fix some arbitrary threshold, which is commonly done right now, you can see for library size, you're going to be excluding a lot of white matter in layer 1, but for mitotic percent, you're actually going to be excluding a lot of, say, layer 3. And so looking at this across all your samples, you're indeed going to have bias in essentially the data that you are throwing out. And you're going to be throwing out a lot of spots that you shouldn't. But now, if we look at the Z normalized data from the SpotSweeper, it really fix a lot of these distributions within the library size, the meta ratio, and unique genes. And so now we've kind of reduced that bias that we're no longer excluding spots based really on the spatial domain, but also retaining a lot of those spots. One of the cool things about this was that as we were using this method, we were starting to look across data sets, and I noticed this interesting pattern that kept coming up. We saw the same spots were actually found as local outliers across almost every single sample. So this is a few data sets of 12, 30, and 1 sample here. So it's a total of 43 samples. And all 6 of these spots were actually found as local outliers in over half of the samples, some of them in all 43 samples. And so looking into this further, we found that for each individual spot in the Visium capture array, that same spot in that XY coordinate has the same synthetic DNA barcode across all capture arrays. So we thought it may be this DNA barcode that underlied it. So looking into that, we do in fact see pretty strong homology in these synthetic barcodes underlying these spots. And so we speculate that this DNA barcode is based on previous data and previous work. We're speculating that these synthetic DNA barcodes in these spots are actually driving downstream PCR bias, and these are essentially systematically bad spots within the Visium capture areas. Moving on to the regional area. So another unique thing about SRT is that we no longer an additional complication is that now we have these actual tissue sections. So we're taking a tissue section, which is very complicated, very thin. Now we need to place it onto a slide, and that is creating its own artifacts that is unique to SRT, and there's really no methods to find these sorts of artifacts. One I wanted to highlight here today. The group that we work with has a lot of post-mortem brains from humans, and so these human brains are obviously way too large to fit onto one slide, so I have to use a drill to actually cut out the piece of tissue that you want to put on the microscope slide. That drill obviously kind of tears up the tissue on the side, but sometimes that tissue is still, within the realm of what you want to put on the array. And so you end up with this sort of hangnail artifact, as we're calling it. So this is this area here on the side, and we can tell this is a clear artifact because using clustering, we can see the normal layering of the cortex. However, this hangnail artifact is clustering in with the layer 6 essentially from this data. And so it wouldn't be as big of an issue if this artifact actually clustered out by itself and you could just exclude it, but that's not the case. But we noticed if you look here that this artifact tends to have a very low variance, almost as if there's a smudge in the mito ratio. So to address that, we wanted to target this as a way to fix it. And so we use a very similar method where we take the local variance across several different neighborhood sizes. We adjust for the mean variance relationship. We conduct PCA to separate out the 2 clusters, and then we can essentially cluster using k-means. And doing so works out pretty well. Just plotting PC1 clearly separates this hangnail artifact from the rest of the tissue. We can cluster that out, and when we do that, we actually increase the rank of known marker genes for layer 6, where that artifact was previously clustering in with it. Good timing. So this is now, this is currently on Bioconductor and preprint as well. If you want to dive into more details that we have online. Thanks to everyone who's helped.
Yes, well, 2 minutes.
We got one question we'll do online first. So from Vineeth Raguram, can we incorporate the aligned H&E data feature averaging to do this? QC metrics with both the gene and similarity in the H&E or MIF data?
I'm not quite sure I followed the question there. Can we incorporate the aligned H&E data to do this feature averaging QC metrics with both the gene and similarity in the H&E, or I'm not sure that I'm entirely following the question. Currently, we don't have any methods that actually incorporate the H&E data. So for instance, like you can sometimes see artifacts in the H&E. So if there was some way to actually incorporate that kind of visual observation, that would be great. I'm not sure if I answered that question, but I can try to address that on Slack as well.
Hi, this is really cool work, but I am concerned that as you move into other technologies, you might have a little bit of a problem with doing this nearest neighbors approach to your normalization, because post-mitotic cells like neurons are extremely transcriptionally dense compared to their mitotic counterparts. And so what you can actually end up seeing is if you had a neuron surrounded by other non-neurons, you'd see very different kind of spectra. So do you have any kind of plans for normalizing that out of this kind of normalization scheme?
Yeah. So that's a great point. So we haven't really delved into the single cell technologies for that reason. It's much more complicated. You said you actually have different cell types sitting next to each other. So how do you really tell what's a bad cell type from another? And so my best guess there, again, I haven't dove in, but there's also different QC metrics that are being used for image-based. You have negative control probes and such. So my guess is that there are probably other metrics that we're going to need to use to actually tell, for example, maybe bad segmentations, if you will, because that's also not a perfect process. So I think that there are ways that we can certainly get at that, and I think improvements that we can make based on a similar system. But I think it will be a matter of the fact that you don't even have mito genes in the image-based system. So it will take some thinking about like what are even the best QC metrics for these methods, and then trying to figure out what's the best algorithm to actually solve these issues also.
Yeah, so it is really cool though that you don't really tend to see that in the data that you've looked at, because I figured that might have been a problem here. But
yeah, thanks.
All right, one quick last one from online, and then Boy, I'll ask you maybe if you could speak to him at lunch. Hopefully it's quick. Will this work for Visium HD?
So we have not tested that yet, mainly because getting Visium HD into the SPE objects, which this is based on, has not perfected yet. There's nothing that should hold it back. So that's something I'm definitely wanting to look at. One thing with Visium HD is the resolution can change. You could have 16 micron resolution, 8 micron resolution, or segment and get like the individual cells. So the actual parameters and the size of the neighborhood will definitely need to change based on that. But something we're definitely interested in doing.
Thank you. Okay, next up is Charlotte Hutchings, using high-throughput spatial proteomics as a platform to elucidate protein relocalization events during viral production.
Hello, everybody. I'm Charlotte, and I'm currently a third-year PhD student over at the University of Cambridge. And today I'm going to tell you a little bit about what our lab does. We focus on subcellular proteomics, so we're not talking about tissue level anymore. And I'm going to tell you about my project using subcellular spatial proteomics to try and identify moving proteins in cells when they're producing virus. So I'll first talk about my biological question and the motivation behind this before explaining the experimental methods and then the bioconductor packages and the pipeline we use to analyze the data. So I'm interested in gene therapies, specifically gene therapies that use adeno-associated viruses, or AAVs. And these are viruses that we take their genome out and we put therapeutic inside of them, and then we give those to patients. Sorry. So the way we manufacture these AAVs is by doing a transfection of a HEC293 cell. We give it 3 separate plasmids. The first one, shown here in purple, contains those normal AAV genes that encode for proteins required to make that viral shell. We also have adenoviral genes because AAVs themselves can't actually replicate in the absence of an adenovirus, hence the name.
There we go.
Then we have a third plasmid which has that therapeutic gene in the
Yeah, I
all of the
messaged him in the group.
plasmids, all of them expressed in these HEC293 cells, but only the therapeutic plasmid, shown there in turquoise, will be packaged into the final virion because it has the additional packaging. So my overall PhD project is to try and characterize these HEC293 cells producing AAVs, and the motivation behind this is to try and find targets to hopefully engineer a more productive cell line. And the specific question I'm going to focus on today is whether or not any of those host cell proteins are moving around within the cell during the viral production. So in our lab, we do subcellular proteomics, and we've developed a method that we call LOPIT-DC. So what we do initially is we lyse the cells, we break the plasma membrane, but we do this in quite a gentle way such that we don't break the intracellular structures. We still have intact organelles. We then take this lysate and we do a series of centrifugation spins at increasing speeds and for increasing durations. So I will do one spin and then take the supinate and then spin it again for longer and faster. And what happens during this process is that the organelles will pellet out based on their density. So we'll have denser organelles such as the nucleus pelleting in the first couple of fractions, the mitochondria, and then all of those free proteins left in the cytosol, they won't be present until the later fractions. So once we've done this, we use standard liquid chromatography mass spectrometry to go ahead and identify and quantify proteins in each of these fractions. And this creates the matrix that you see there on the right. And after normalizing to account for the total protein abundance, what we end up with is what we call abundance profiles, where each protein has an abundance profile across the biochemical fractionation gradient. And the way in which this actually gives us subcellular information is that we expect proteins that are in the same space in the cell to co-localize and therefore co-fractionate, and they should end up with pretty similar abundance profiles. So in our lab, we've developed bioconductor packages to analyze this type of data. The main ones are the Proloc family of packages. And the first thing we do is we annotate our data set with marker proteins. So these are proteins that we know their subcellular localization, we know where they reside, they don't move under any condition, and we annotate these within our data. So what you can see at the top here is 2 examples. You can see the profiles of the mitochondria in green and the cytosol in light pink. And all of the proteins in these compartments should have similar abundance profiles, which when we then use dimensionality reduction means that they would cluster together on the PCA plot that you see here. So we then use these marker proteins and their abundance profiles as inputs and training data for semi-supervised machine learning algorithms, which are also stored within the Proloc package. And this allows us to compare the abundance profiles of our unannotated proteins to those marker proteins in order to try and work out where they would be residing within the cell. So for my PhD, I've done this 2 times over. I've done it on some control cells, the normal HEC293 cells, and then also on AAV-producing HEC293 cells. So once I've created my static maps, the next thing is to then extend to see what is differentially localized. I can compare those classifications between conditions and see which proteins are classified as being in different organelles when my cells are producing AAV. And in this case, I won't go into too much detail, but what we saw was quite drastic rearrangement of the nucleolus, which has been confirmed using the electron microscopy. You can see there on the right of the screens, quite drastic differences. We also are able to observe the formation of stress granules with lots of proteins moving around there, and also rearrangement with proteins moving through the secretory pathway from the ER and the Golgi. However, this approach itself is limited by simply just comparing those 2 separate classification lists. It's a qualitative approach. We're not going to get information about the actual probability that a protein is differentially localized. Previous approaches to solving this problem have simply used delta profiles. So they'll see whether a protein has a shift in its profile, and then they'll look at the reproducibility of that shift. But that is limited where we have changes in cellular morphology. So in my case here, in viral production, the cells changed quite a bit, which means that the actual organelles may pellet differently, even though proteins aren't moving between them. So in the last couple of years, we had a PhD student in our lab called Oliver Crook, who developed the Bayesian analysis of differential localization experiments, or BANDL. And this is now its own bioconductor package. And here we're able to model 2 different conditions together. We give it both conditions. It's able to classify, but also give us uncertainty estimates for whether or not a protein is differentially localized between our 2 conditions. And this is based on semi-supervised MCMC framework. And hopefully that's just an introduction to how we are using subcellular spatial proteomics. Lopit DC can characterize the localization of a protein, both in a static sense and in a dynamic framework. We've developed analysis pipelines for this. We also have several F1000s to walk everyone through the process of doing this. We use Q features as the structure for our pre-processing, then Proloc and BANDL. And in my PhD, I've been using this to try and work out what's happening inside those AAV-producing HEC293 cells. So with that, I want to thank everyone back in Cambridge and my AstraZeneca colleagues, as well as Bioconductor, for giving me the platform to speak today.
Hello, thank you for the great talk. So I have a question about, because this is LC-MS based. So I'm wondering, like, do you have a lot of missing values in your data set? And also, like, are the missing value patterns consistent across different substructures? And are you doing imputation? Since I see a PCA plot, so I guess you're doing, if yes, so what imputation are you doing?
So this method's been around for quite a while. So it was developed when data-dependent acquisition before DIA became popular. And we actually make use of tandem mass tags, which is the T in Lopit DC. And the benefit of a tandem mass tag, which is normally a disadvantage, is that you usually get a very small value of carryover where there should be a 0. So actually in these data sets, we don't really end up with more than like 1% missing values. So we tend to just remove any protein that has any missing values. We don't really lose a lot by doing that. And then when we do that normalization, those tiny values just become basically a flat profile anyway. So actually where the difficulty is now, we're trying to translate this into a DIA version. And now the missing values are becoming more problematic, but we're still trying to work out the best way in the workflow to deal with that.
Questions? I don't see any online. Okay,
Cool.
thanks, Charlotte. Okay, our final speaker of this section of the session is context is important. Identifying context-aware spatial relationships with Kontextual by Farhan Ameen.
Alrighty. My name is Farhan. I'm a second-year PhD student, and I'm obsessed with spatial analysis. So at its simplest form, we can describe the relationship between two cell types as either dispersed or as localized, where they're sort of intermingling as a sign that they may be interacting or communicating. However, this is not as simple as it seems. Take this toy example right here, where we have two cell types which here appear randomly distributed within the image. I don't have enough evidence to say they are localized, nor do I have enough evidence to say they are dispersed. But if I were to zoom out of this image, now these two cell types appear dispersed from one another. One's in the top left-hand corner, one's in the bottom right-hand corner. If I were to zoom out yet again, now these two cell types appear localized in the bottom right-hand corner of the image. So clearly, the way that we are quantifying these tissues has a big implication on the spatial relationships that we quantify, which is actually a problem because that means that spatial relationships found in one patient image may not be transferable to other patient images, which highlights a critical barrier to identifying consistent spatial biomarkers across diseases. And so one thing we set out to ask is, can we find a context in which we can evaluate these spatial relationships that aren't affected by the way that we image these tissue regions? And one thing we came up with was using the cell type hierarchy. So as I'm sure everyone knows, the cell type hierarchy is when we have our homogeneous populations of cell types at the bottom of the tree. And as you go up and stratify up the tree, that represents parent populations with more shared generalized functions. And so what we're doing here is we're taking a relationship between two cell types, in this case, red and blue. And as we can see, these two cell types appear dispersed. And we're incorporating this parent information into this cell type relationship. And so now you can see that relative to the spatial behavior of red's parent population, red appears much more localized with blue relative to that parent population. And so I hope you can appreciate that no matter how I twist and turn this image, if I zoom in or out, red is always going to appear more localized to blue relative to its parent population. And so we think we can use these parent populations as a context in which we evaluate our spatial relationships that aren't affected by these imaging biases. So that's how we choose our context. Next, how do we calculate these spatial statistics themselves? Well, one thing we use is the stock standard L function, which is a really popular spatial analysis tool. And what the L function does is it forms a radius around one of our cell types, in this case red, and counts the number of blue cell types within that radius. It then goes around and does this for each of the red cell types and then takes an average. And this gives us an average level of association between red and blue. And what we've done with the L function is just augmented the weights slightly to incorporate this new parent context information. So let's look at an example in practice. Here we have a triple negative breast cancer image. And in this image, we have immune cells in black, tumor cells in light gray, and a subset of tumor cells labeled p53 positive in green. Now, if I were to characterize the spatial relationship between these p53 positive tumor cells and these immune cells using the L function, you can see that we get a negative relationship over the range of radii that we measure. And what this is telling me is that the p53 positive tumor cells and the immune cells aren't mixing whatsoever. These two cell types are dispersed. However, when I use my method Kontextual, you can see that we start to get some positive association over some range of radii. And what Kontextual is telling me is that relative to the spatial behavior of all the tumor cells in the image, these p53 positive tumor cells appear much more localized to those immune cells, which we can confirm by eye, where we see that these green cells appear much closer to that tumor immune interface relative to those gray tumor cells. Now, one key thing to highlight here is that a discrepancy between the L function and Kontextual, in this case, one is negative and the other is positive, can highlight relationships where adding this context information changes our spatial conclusion. So what we can do is we can calculate all pairwise relationships within the data and plot the L function and Kontextual metrics. And in the red squares here are where these two metrics disagree with one another. And so here I've identified a relationship between these tumor cells and CD8 T cells where the L function and Kontextual disagree. And having a look at the representative image, we can find that we have tumor cells in black, CD8 T cells in red, and immune cells in gray. Now, the L function, which characterizes the relationship between CD8 and tumor, is negative, which states that tumor and CD8 T cells aren't mixing, as the majority of T cells lie outside that tumor region. But what Kontextual is able to identify here is that relative to the spatial behavior of all the immune cells in this image, CD8 T cells appear much closer to that tumor body. And in fact, we do see some CD8 infiltration into the tumor body. So what Kontextual is able to pick up is these subtle CD8 infiltration relationships that normal spatial metrics cannot capture by themselves. And so one thing we did was we calculated this CD8 tumor relationship across all the images within the cohort. And what we found was that when we do survival analysis, the Kontextual metric is able to separate out survival outcomes much better than the L function metric for this example. And so what Kontextual is saying here is that an increase in CD8 infiltration into the tumor body leads to better survival outcomes overall for these patients, which is consistent with what we see in the literature. So overall, just to conclude, one thing I learned from this project is never take your results at face value, because something which seems very obvious at face value may not necessarily be true when you put it in context. And with that, I'd like to acknowledge everyone who's worked on this project, as well as plug in our package Spatial on Bioconductor. Thank you.
Very nice talk. Got a question. How small of a population can you go as far as being able to recognize it on the different parameters? Like, does it have to have a certain number of cells to be actually found out, or could you have a smallest population setting?
So the number of cells obviously makes a big impact. If the cell population is too sparse, you sort of get really unreliable spatial statistics. But we have had scenarios where we go into really finely defined populations of cells, looking at even their states. I think it's just something you have to play with and see. But what we found usually is when the parent population is well defined and there's a lot of cells within that parent, then usually the statistic is fairly robust.
So maybe more like a comment. So yeah, it's great work. It's really cool. You brought up the spatial point pattern analysis tradition. So like there, the observation window is very important, and having different observation window will drastically change the results for like K function, L function. So here my question when it comes to contextual is that so number one, so what if so what if you have like a large piece of tissue? For these days, we are really emphasizing like scaling up the single cell resolution technologies to like even to like a whole organ level in like mice, for example. So what if you have like a large piece of tissue and so you have regions in that tissue where like two cell types co-localize in another region where they don't. So and also like here that leads to my comment when it comes to the observation window. So like in geography, like I think often the extent of the spatial region of study is considered beforehand. So like would consider like only doing your analysis like on a part of that like whole organ level tissue instead of another. That could be like an art. Yeah.
That is a great question. I guess spatial technologies haven't gotten to the point where we can capture that much area yet, but we have applied this on Xenium images where we've clearly seen very different localization patterns based on the different regions. And I think the approach in that was you can still characterize like different tumor structures based on the certain typing that like different tumor structures may not be exactly identical. And so in that case, we've treated different tumor structures as different kind of observation windows, if that makes sense. Like one type of breast cancer tissue might be DCAS1, whereas another might be an invasive tumor sample. And so we treat those sort of differently. But yeah, I don't have a good answer for that question, but it is a great question.
Yeah, it's a decent answer. I think yeah, but I mean like now we also have like whole embryo sections like from mice, so like that could be relevant. Like
Well, good. So I know there's more questions here in the audience, but we need to move on. So maybe all the spatial people can sit together at lunch. You all can discuss. There was lots of good talks. So help me thank all our speakers today from the session.